This is the third Citizen Science Lab Procedures video. Most phage can switch between two different lifestyles. A phage is a bacterial virus. During one of these lifestyles, called lysogeny, phage genomes are incorporated into the actual bacterial genome. Each time the bacterial cell reproduces, the phage genome gets copied and passed on as well. In this case, the actual phage viral structures are not made, but rather they lay dormant until the surrounding environment changes. Lysogenic phages can also encode for antibiotic resistance genes and can pass on this property to infected bacterial cells. The lysogenic lifestyle can shift to a second lytic lifestyle, which is the type you will look at in the lab today. In this case, instead of the phage genes only getting copied, they are transcribed and translated, producing many viruses that can fill the cell, causing its lysis, which means it's bursting, and spread to infecting surrounded cells. Something to note real quickly here is just simply that when you see this color change here in the DNA, okay, do you remember what this DNA ring is called? Plasmid, okay? So when the color changes in a picture, it's showing you the incorporation of the phage DNA into the bacterial DNA, okay? And then up here in this part of the drawing, you can see all these parts, right? We were talking about how in one life cycle, in the lytic cycle, these parts just kind of sit here waiting, all right? So two parts, the DNA work and then the actual work of reproducing viruses. Don't have an image of the very first thing, but you're going to melt the top agar in the 100 degrees Celsius water bath. Once melted, move it to the 55 degrees Celsius water bath for at least 10 minutes. It must cool from 100 to 55 degrees Celsius or the heat will kill the bacteria. Okay, this part has already been done. So your main job here is to make what's called a serial dilution. Serial just means a sequence, and dilution, you should know what that means, and we will talk about it quite a bit. Okay, so we're going to make serial dilutions of phage stock T4 as follows. You're going to label the sterile 5 ml culture tubes that have blue caps. Label the tube, not the cap. Okay, the cap, just like the lid of the agar plate, can get separated from the important part, which is the tube. Okay, you're going to label 7 of these. And remember that the microcentrifuge tubes are just really little tiny tubes, okay? And the agar plate is a big plastic circle or square plate. And this particular kind of tube is called the culture tube, okay? Once you have all of the tubes labeled, you're going to add 990. Be careful because we're going to see 900. So the first thing is add 990 microliters of broth to tubes labeled 1 to 3 and 900 microliters of broth to tubes labeled 4 to 6. Add 10 microliters of your phage stock to tube 1. Mix thoroughly but gently, and then change the tip. Remove 10 microliters from the tube 1 and place it in tube 2. Mix gently and change the tip. Remove 10 microliters from the tube 2 and place it in tube 3. Mix gently, change the tip. Remove 100 microliters from tube 3 and place it in tube 4. Mix gently, change the tip. You're going to keep doing this with 100 microliters until you get to tube 6. Prepare tube 7 which is a sample without phage, by adding only 1 milliliter of tryptone broth and no phage. So remember we talked about how tryptone broth was Gatorade that wasn't quite as good as LB broth. Label your tryptone agar plates 1 to 7, including your initials and date. Pour 4 milliliters of top agar into each of the 7 prepared sample tubes. Immediately pour the mixture onto the agar plate and swirl the plate gently. Do this 1 tube at a time to reduce the cooling of the top agar. To reduce contamination problems, start with the most dilute phage samples, then proceed to the less dilute ones. In other words, start at tube 7 and go to tube 6. After the plates solidify, invert and place them in the incubator. Incubate them overnight. Let's take a quick step back and just think for a second what this dilution series is. So we already said a series is a sequence, and I said you probably know what a dilution is already. Okay, now let's go over that. So let's take a look here at the bottom right here. You have 2 tubes of the same volume, okay? So I didn't draw it perfectly, but you can see that, right? They're the same tubes, and this line here is trying to indicate to you that it's exactly the same volume. Now, if you count the number of X's in this tube, there are 10, okay? In the first dilution, we only move one of them over here into the same volume, okay? What you need to understand is that the dilution series is a practice in reducing the concentration of solute in the solution, okay? The solute is the solid part and the solution is the liquid part, okay? And then we'll go over this in class quite a bit, but hopefully your eyes have just been resting on it while I've been talking. As you might remember from math in high school, the basic point here is that we have a dilution factor and it's in tens, all right? So all of these here represent each one of those test tubes and each one of the times that we mixed one to the other, the blue culture tubes, okay? In the end, what we find is that all we have to do to figure out this math is to count up all these zeros, all right here. And then we can make a really pretty factor of 10 using this thing, which is called a superscript. And the superscript simply tells us how many zeros to add to this nice, neat number. If that number in the superscript is negative, it means move the decimal point to the left. So the number is much less than 10. If the number is positive, you move the decimal place to the right, so the number is much larger than 10. So understand that with each dilution, are we making it smaller or bigger? Okay, that's what you want to think of. Is it bigger or smaller? I'm not going to tell you the answer right now. You'll see the number and you should be able to figure out if each successive dilution results in a concentration that is greater or less. The next time you come into the lab, you're going to count the plaques on the plate. A plaque is a clearance, okay? When it looks all fuzzy, like these ones do over here, okay? This is called the bacterial lawn. That fuzziness is 100% covered with microbes, okay? But a plaque is a clearance. The plaque is the virus, and it's explaining to us how much space was contaminated such that the bacteria could not grow in there, okay? Now, over here we see it's really clear. So here there's too much clearance. There's nothing to count. There are no microbial communities. I mean, there are some very tiny ones you can see here. But then over here, there's too many colonies over here. So somewhere in the middle is going to be the perfect situation where you have just enough colonies to count them, okay? And you might see here that we actually drew on the lid with a Sharpie to count them. You don't want to do this right away because you want to be able to see them, but that's a great way to count them. You can always remove the Sharpie with alcohol.